Thank you so much for the introduction. And like Mata said, my talk is about histological validation of rPCA-based myelin water imaging. And we're doing this in a panel of variably hypomyelinating mice that I will describe to you shortly. So a little overview of my talk. I'm going to give a brief introduction to myelin water imaging. And I'm going to talk about a new processing technique for T2 star-based myelin water mapping. And then I'm going to talk about how we're going to leverage a panel of hypomyelinating mice to calibrate these measurements. And then finally, we're going to take a look at the integration of this myelin water imaging technique into the MRI G ratio framework. So here's your myelin water imaging. Broadly speaking, what MWI seeks to do is to non-invasively measure the quickly decaying T2 or T2 star component corresponding to myelin water. So if you look at this little plot over here, this is a spectrum of logarithmically spaced T2 star values. And on the y-axis, we have the spectral density. And if we have record a reluxometry experiment and we do some processing, we can claim to split these non-orthogonal exponential components into their corresponding amplitudes. And MWI is typically done, like I said, in using either T2 or T2 star relaxation. And traditionally, it's been done with the multi-echo spin-echo experiment, going back to Whittle and McKay, other experiments by Eva Alonso Ortiz here at the MNI, lots of work at UBC leverages this T2-based technique. And it's also recently been gaining traction doing it with T2 star instead. And the advantages of using T2 star as opposed to a multi-echo spin-echo experiment is this quicker readout, lower SAR, a little bit noisier, but overall a full brain coverage for a human or a preclinical experiment can be done more expediently using 3D T2 star based imaging as opposed to 2D multi-slice based multi-echo spin-echo. So once this experiment is done, in order to actually get your myelin water signal, people have traditionally used either non-negative least squares, which is where this plot comes from, which basically projects the relaxation signal onto, like I said, non-orthogonally spaced, non-orthogonal logarithmically spaced exponential functions. So not exactly like a Fourier transform, but just in the real domain. Or people have before also fitted this three-pool model of three-component, three-pool model of relaxation using the magnitude data or using the complex data where phase is incorporated. And then finally, once the splitting is done, your myelin water fraction is computed as the ratio of the myelin water to the myelin water plus the intracellular and extracellular water. So we term that the myelin water fraction. And basically, it's essentially measuring the amount of protons that are trapped between these lipid bilayers that have this characteristically short spin-spin relaxation time. And it's separated from these intracellular and extracellular pools. So recently, our colleagues in Korea have come up with this very, very interesting technique called blind source separation with robust principal component analysis. And basically, BSS rPCA, as I'll shorten it, is a series of matrix operations that extracts the myelin water signal directly from the gradient echo data, as opposed to doing this ill-posed and not ill-posed, basically the version of fitting where you would have more points than data. But basically, rPCA instead separates its T2 star weighted gradient echo signal into what are called unit rank components. So unit rankness in this case means monoexponentiality into these two components, this L2 unit rank component and L1 unit rank component, which represent the myelin and intracellular and extracellular components respectively. And it's completely model free. So we're not fitting three-pool models. We're not doing an NLS. It basically extracts the myelin component using matrix operations, and it does this very, very quickly because it can be implemented with linear algebra primitives in your language of choice. Unit rankness, in this case, monoexponentiality of the corresponding signals, is enforced using Hankelization, which is a technique from matrix signal processing that essentially makes things behave, in this case, monoexponentially. And in addition to splitting this total MGRE signal into univariate components, it also performs a denoising component because it sequesters artifactual components of the signal into a sparse component. And what this looks like numerically is essentially we have this convex optimization problem where J, which is a function of these two components, L1 and L2, as well as S, where essentially we try to solve this problem by, from M, removing L1, L2, and S in a patch-based method, which is this RR operator on the Hankelized components. And the back end of this has quite a few more calculations on it, but the full derivation is available at this link over here. And then finally, once you perform this, you can compute your Myelin water fraction from this L2 component over the sum of L1 and L2 at the first echo time. So effectively, you're regressing to the relative densities of these two components at the first echo. So what this looks like pictorially is the following. So if we look at panel A, this is our MGRE signal for a mouse, and we can see that it experiences T2 star decay, and after it's split into L1 and L2, we can see that the L1, the slowly decaying component, behaves relatively similarly to the original signal, but the L2, this quickly decaying component, it decays quite quickly, and some noise is sequestered into the sparse. And if you do a mono-exponential fit to these respective components, we can see that the proton density of the MGRE original signal is pretty uniform. But when we look at the slowly decaying component, L1, we can see that now there's hypointensities associated with this white matter skeleton corresponding to the mouse anatomy. And we can see that the L2 component essentially produces this skeletonized structure of white matter. And if we look at the actual T2 star maps outright, and bin them in a histogram, we can see that indeed this gray MGRE signal is split into these quite nicely defined slowly decaying and quickly decaying components. And then finally bringing it back to C, if you just pick a random voxel in this case the cerebellar white matter, you can see that the time course of the signals also makes sense. They're nice and exponential. L2 decays faster than L1, and at later echoes, L1 and the original signal M converge, indicating that essentially it looks at a glance like we're extracting some quickly decaying component here. So basically, with rPCA out of the way, I want to introduce these mice that I was telling you about earlier. And our colleagues here at McGill, so Human Bagheri, as under the supervision of Dr. Alan Peterson, basically surmised that if you take your MBP gene in a mouse and you knock out, using CRISPR, one or more of these M1, M3, and M5 transcription enhancer domains that are upstream of the MBP gene, the actual, exon that codes for the MBP gene, you can basically knock down MBP transcription, and then obviously, as a consequence, translation, and as a consequence, myelin basic protein elaboration. So they were able to do this, produce a whole panel of different mice that have these characteristics, where if you quantify MBP expression using reverse transcriptase, you can see that these progressively increased knockouts have progressively less and less and less MBP mRNA. And they also produced this other very interesting mouse that actually has an exuberance of MBP mRNA that's transcribed, which we will call the knock-in. So for the purpose of this talk, to keep things short, instead of using these somewhat cumbersome formulgenic names, we'll just call them the knock-in, which is an exuberance of MBP mRNA, wild type, and then single, double, triple knockout, as well as the shiverer, which has a total MBP gene ablation. So it's important to note that these mice aren't a model of any specific disease. They are essentially, for our purposes, a very, very, very fancy imaging phantom that corresponds to progressive hypomyelination, rather than the toxic demyelination that you can observe in a Cooper zone animal instead. So let's get down to the MRI bit. This is our acquisition for our gradient echo. So the key parameters here is that we image these mice at P30, so they're adult-ish, but not fully adult, meaning that they're still in the process of postnatal myelination. Our FOV was, sufficient to accommodate the size of an ex vivo mouse brain. Our spatial resolution was 100 microns isotropic, and critically, our TE, our first TE and our delta TE are 2 and 2 milliseconds in 24 echoes with a TR of 100 milliseconds, relatively low flip angle of 22 degrees, 18 signal averages, and to do one brain on our Brooker PharmaScan 7 Tesla takes give or take 8 hours. No, sorry, 6.5 hours. And basically, we reconstructed these brains with a inverse Fourier transform, and we corrected for some T2 star phase-based jitter, which Dr. Rudko will explain in his talk. And then finally registered these things to the Sick Kids template and parcellated it with corresponding Allen Brain Institute template atlases. And on the right-hand side is what this looks like. So this is our template. And this is an example of a wild type and a severely hypomyelinating mouse, a double knockout. So you can see that, first of all, both have pretty good spatial correspondence to the template. And in addition to that, just at a glance of the signal averaged MGRE, you can see that there's a considerable diminishment of white matter, gray matter contrast in the double knockout as expected. And moving on to these quantitative myelin water fraction maps, we can see that there's a pronounced MWF hypointensity exhibited globally throughout the whole brain when compared with a wild type animal. So the ROIs that we investigated are ones that are prominently delineated, corpus callosum, everybody's favorite white matter tract. In addition to that, the anterior commissure, internal capsules, as well as the somewhat, I think, neglected and understudied mouse cerebellum. So we just picked ROIs that spanned the entire rostral caudal axis so that we can kind of sample what our myelin water imaging method is doing throughout the whole mouse brain. And this is just a cool 3D render from Fossilize that I thought looked nice. But this is on the right-hand side a whole MWF map of the brain, where on the left-hand side this is our template atlas and the ROIs that we use to parcellate everything. So the point that I'm trying to drive home here is that we didn't just kind of like stick our finger at one small spot in a white matter tract. We're parcellating out the whole thing. So it's a lot of signal averaging over a large space. So this is a bit of a busy figure, but basically, if we go and image our entire mouse cohort, in this case, 23 animals, we have 5 wild types, 6 single knock-ins, 3 double, triple knockouts, and 3 shivers, as well as our putatively hypermyelinating mouse. So we image everybody, we put everybody in template space, we compute our myelin water fraction maps, we parcellate, and then we do essentially an ANOVA as a function of genotype. So the legend is over here, our mouse strains. So basically, the darker it is, the less MBP mRNA, and red is highlighted as the knock-in animal. We can see that there's a statistically significant difference between the wild type and these progressively more hypomyelinating animals. And interestingly, we actually don't observe a statistically significant difference between wild type and the knock-in, suggesting that there's some kind of post-transcriptional mechanisms at play that don't allow you to over-elaborate myelin secondary to the saturation of this MBP mRNA signal. But more interestingly than this ANOVA on the left-hand side is if we drop the knock-in and plot our MWF values versus MBP mRNA on the x-axis, we get these really, really nice regression plots where we can see that our computed myelin water fraction is quite a robust linear function of MBP mRNA. Not myelin basic protein, but rather the mRNA, suggesting that from transcription to translation to myelin elaboration to our myelin water fraction sensing method, MWF is closely related to this parameter that we can control, which is quite a neat result, I think. So we took this a step further and we tried to get some additional ground truth metrics of myelin. So in this case, we took 5 additional brains, fixed them, coronally sectioned the whole brain, level matched them, and then stained them for myelin basic protein, as well as light chain neurofilament. So MBP would be our kind of ground truth for our MWF, and LNF could be are essentially a complement to a control experiment to make sure that our myelin decreases in our myelin signal correspond to decreases in myelin rather than axonal loss. That could be, an idiosyncrasy of the mouse model we're studying. And we got a whole bunch of these coronal sections, level matched them, and then we used a color deconvolution algorithm to just pull the intensity of the brown stain. So for those of you that are unfamiliar with this type of chromogen, this is DAB, diaminobenzidine, and the idea behind immunohistochemistry is that you have an antigen, in this case, myelin basic protein. You target it with your primary antibody, which is sorry about that, which is specific to your antigen, your secondary antibody crossing to your primary antibody, and then in the presence of hydrogen peroxide, the secondary antibody essentially facilitates the conversion of DAB into this brown stain. So the more brown it is the more myelin basic protein there is in the stain. So again, collapsing all this information into one figure, we looked at these same ROIs that I showed you for the 3D parcellation, but we took coronal slices from a template atlas, parcellated our same white matter tracks that we were studying earlier, and we developed an in-house registration pipeline to map these raw immunohistochemistry images to our MRI template. And because our MWF maps are already in our MRI template space, we can make a quantitative comparison between MWF and what I call VDAB, this virtual diaminobenzidine image, where we, like I said, we deconvolve out this brown stain and apply a color map to it. So the thing that we're looking at is essentially this blue and this red. And we make the argument that if you use these parcellation masks in 2D space as a function of our progressively hypomyelinating genotypes, we can see that our MWF maps and our VDAB maps align quite closely, which is rather encouraging. And in order to quantify this, we apply these parcellation masks, and we can essentially do a similar linear regression as a function of genotype, where if we normalize everything to the wild type, which is presented in gray, so I just took my VDAB and my MWF values, and I just normalized them to the spread of the wild type. So relative to the wild type, we can see that, using the same color maps as we did before, the darker it is, the less myelin it has. We can see that they, A, cluster out really nicely as a function of genotype, and B, we can see that we have, again, a rather robust linear relationship from MWF now to our ground truth, our actual myelin basic protein. So that's further encouraging that when we're trying to do this myelin water imaging technique that, it essentially tries to split the protons that reside in these intralamellar spaces and therefore have this characteristic short spin relaxation time, we can see that we actually quantify the myelin, or the myelin basic protein itself, they are also linearly related. So that's also encouraging. And we decided to also complement all of this with a diffusion study. In this case, we used diffusion weighted gradient and spin echo sequence that was implemented here at the MNI. Our spatial resolution was 150 microns, and one of the critical parameters in our diffusion acquisition is our this very long TE over here. So it's 35 milliseconds, and so that's about almost 2 decay constants of myelin. So we can say that although there's T2 waiting on our diffusion, we hope that with this long echo time that any contributions from a myelin signal that can leak into the diffusion have been removed. And anyway, these are our gradient directions. And then we took our diffusion data and we hit it with the naughty model, which basically separates intracellular versus extracellular components. And that's, the naughty model is the naughty model. It's quite a broad field of literature just right there. So I'm not going to get into those details, but the parameters that we used were those that were described by Wang for specifically ex vivo glutaraldehyde fixed mice. So we did take into we did carefully consider the fixed parameters that we implemented for naughty, and they differ quite significantly from those that are implemented for human in vivo, with a warm, living, breathing human. This is a room temperature fixed mouse brain. So we did consider that. So unlike our MWF results, if we, apply these same parcellation masks to our diffusion experiment, register it to the template, parcellate it, and spread it like this, unlike our MWF, our AWF, our axon water fraction derived from naughty, which is, we're using it as a surrogate of axonal density, it does not behave in the same way. We observe very few statistically significant differences from wild type as a function of genotype, and our corresponding regression plots also, while they are, a few of them are statistically significant, their slopes are essentially within experimental error or experimental uncertainty from 0. So in this sense, we can say that changes in our myelin water signal originate from decreases in myelin rather than some sort of, metabolically linked axonal loss that could also be happening with this hypomyelination that we observe in these mice. So that's cool. And we did the same thing with LNF again. So we went there as well. We co-registered our light chain neurofilament immunohistochemistry to our template, and we did a quantitative comparison of axon water fraction versus our light chain neurofilament as a control experiment for axonal density. And again, we apply the same parcellation masks to our LNF. And unlike our MWF experiment, we can see that, first of all, these scatter plots where everything is again normalized to the wild type of AWF on the y-axis and light chain neurofilament VDAB intensity on the x-axis, we can see that they A, don't cluster out by genotype, and B, they don't have any meaningful slope associated with them. So while LNF is not necessarily a direct surrogate for axonal density, it complements our diffusion study in that it just further reinforces this notion that we are not, getting a lot of axonal loss over here. So our signal is tightly localized to our myelin component. And so armed with this information, we decided, hey, why the heck not? We can compute G ratios according to the Stickoff formalism and supported with biophysical modeling that was introduced by Captain West under the supervision of Mark Does. And this is a nice figure from Siwish Mohammadi that describes kind of like this G ratio formalism, whereby if we have a big, MRI imaging voxel, we can make some geometric arguments about how to compute the axon area weighted mean G ratio of all fibers within an imaging voxel. So, we apply these biophysical models and we compute a G ratio by co-registering our AWF with our MWF as well. We scale MWF to MVF, and we compute axon volume fractions by scaling our axon water fraction by 1 minus MVF. So we can then produce these quite nice looking G ratio maps. And we can see that as a function of increasing hypomyelination, we can see that our G ratio contrast is increasingly, obliterated in these animals. So if, the lower the G ratio, the more myelin essentially, under the assumption that axonal density stays the same. So we can see that, all of your white matter tracts that we're familiar with become less and less and less pronounced, suggesting that our G ratio is increasing because our myelin is decreasing. So again, let's parcellate all these maps and relate them to MBP mRNA. And just to summarize here, we can see that our G ratio as a function of our MRI-derived G ratio, as a function of our MBP mRNA, also exhibits a quite nice linear relationship in all white matter tracts studied. So that's about it for me. To summarize, I showed you guys this rPCA-based method for myelin water fraction mapping. It's just a computational method. It's can be implemented with simple matrix operations. It's computationally expedient, takes 5-10 minutes for a whole brain, and it performs denoising. And it's, clearly it's sensitive to myelin content as we've belabored in this talk. And complemented these experiments with diffusion and biophysical modeling in terms of axon water fraction. And then we put these things together to try to compute the G ratio using these techniques for the first time. And further histological validation of this is in progress. We're doing some scanning electron microscopy experiments on these white matter tracts. So hopefully stay tuned. We'll have additional results to support that. So I'd like to obviously acknowledge all of my colleagues at the Shark Tank, so the MNI BIC MR unit, as well as Dr. Alan Peterson and Dr. Kuman Bagheri, who developed this mouse, which was the linchpin of my entire project, as well as the folks at the GCRC Histology Core for producing the histological slides for me, as well as finally our funding from HBHL, NSERC, MS Society of Canada, and FRQS. Thank you so much.